We're here. Okay, so it makes me so happy seeing all you guys trying to, or not trying to, but collaborating in the chat before I even get here. It is awesome. So yay for you guys. Y'all make me so happy. Oh, hi. For those of you who have not been here before or who don't have me like in class or something, my name is Jessica Nadsum. I am a 5th year AP Bio teacher, so I like to think that I kind of sort of know what I'm talking about. And I am here today to talk to you about the Transformation Lab. So, whoop, 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 whoop. I'm going through the people, and I see so many people here who I know or who have been here before, and that makes me so happy. Yay! I'm so glad y'all are here. Okay. Let's go ahead and pull up the stuff we need to do the stream. So I hope you guys are having an awesome day, evening. Obviously, things have been a little crazy lately, but the important thing is we are all taking it one day at a time and doing the best we can. So what y'all really need, to be perfectly honest, out of this time, is some AP Bio help. I believe it was Friday, it was announced what they were doing with the AP Bio exam. So that has changed since I was here last week. I did this last week and it was a little different then. But, you know, you roll with the punches. Woohoo, what can you do? So one of the things that they did officially confirm, and I saw you guys talking about this in the chat box, is the AP Bio exam is open book. So whoop, whoop. I saw some people were looking at making like info sheets and stuff like that. Totally encourage that. But what I will tell you, as someone who grades the AP Bio exam in the summer, like that's my job. They pay me to go sit in a conference room and grade for 8 hours straight, is they have been very explicit about the fact that while it's open book, there will be no credit for anything content related. Nada. Zilch. They're not going to ask you what's the powerhouse of the cell. And you're not going to be like, mitochondria, give me a point, because they're going to be like, no point for that. This is kind of how they've come around, because like they can't say no cheating, because let's get real. Someone creative out there will be like, hee, but I got away with it. And so it's easier for them and more realistic to say, fine, be open book. But it won't really matter because like you don't get any points for anything that's in the book. The only things that they are grading on is going to be on application. Share the slideshow. Can y'all not see the slideshow? Should be on your screen. So just wanted to point that out there. I strongly encourage you to have info sheets because honestly, just making those is a really good way of studying. But don't rely on them. Just like I tell my students, we met via Zoom earlier and I was like, it's open book. But you still better know what's in the book already. Because if you're running around skimming through your book for every other question, then you are going to waste your entire time on the test and not actually be able to answer anything. And you're not going to get any points for it. So know your stuff. You guys are here, so yay. Kathy, I will share the URL at the end of the lesson. So in the meantime, it'll be up here on the screen now. All right, so without further ado, I do see we've already got some questions. How is everyone's quarantine? Terrific. Hope you guys are doing well and staying safe. Lots of social distancing. I understand a lot of students, though, obviously work during this time, or they may be taking care of family or siblings, things like that. Just do what is safest for you, safest for your family. Take care of yourselves. What is most important right now is that we all remain calm and cool and collected like little cucumbers. So just do that. And then someone else said, whenever I try to watch the previous bio live stream, 2015 FRQ that was live earlier, it says, can't find the event you are looking for. What should I do? That just means that that event has not been uploaded yet. So give it like 24 to 48 hours because it can take a minute to download things from Crowdcast. Especially, I don't know about y'all, but like my internet has been super slow lately. Partially because I think my town had like a tornado hit it a while ago and I think the towers got a little messed up. But also, I think they're just overloaded because everyone's at home on the internet. And so like, it's just using up everything. You went for a hike on Sunday? I'm so proud of you, Rudy Garcia. That is so good for you. Good for you, Rudy Garcia. But yeah, my internet's been super slow. So like I've tried to upload things for my students and it's been really slow, but it'll be okay. So, all right, without further ado, I'm going to take you to the step that you actually came here for. Okay, so we've got this transformation lab. But before we talk about that transformation lab, I have to give a plug. So what I usually assume is that if you're in one AP class, you're probably in like a couple of AP classes. Maybe you're not. That's cool too. But if you are, you should definitely take advantage of different subjects that we have streaming. Mondays, we have English and math. Tuesdays, we have social science. Wednesdays, we have science. Thursdays, we have history. So you should definitely look at taking advantage of those. They're all free, just like this one. Boop, boop. You know, they're all like on Fiveable. We have a group message basically where we're talking about all the things that are changing right now. And we're talking about all of the things we need to do to get you guys prepared for AP exams. So everyone is very up to date, informed, highly qualified. So take advantage of that resource. Most of us are teachers and we really, 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 really miss working with our students. So talking at a screen on the internet is like the highlight of our day because teaching is our job and we can't do it in the traditional sense right now. So, all right, that's my plug. Oh wait, I have another plug. If you're like me, you were born with like a phone in your hand because I'm a millennial, but I'm a late millennial. I was very close to Gen Z. And therefore, I really, 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 really like my phone and I really, really, really like social media. And Fiveable is on social media. They have Twitter and Instagram and YouTube and a bunch of other stuff. So you should definitely follow it because that's when we give updates, like especially with the AP situation, we've been following it really closely. We'll also let you know, hey, this stream is about to happen and da 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 da. And if you're doing cool stuff and you tag us, we may retweet you because like that's all we ever want is likes and retweets, right? So yeah. Okay, that's my other plug. We're already 7 minutes in. I need to move on. Oh, someone else asked a question. What do you think will be on the AP test? I will tell you that by the end of this. How should we study for the exam besides reviewing the content if it will just be on application? We are going to practice application. That sounds terrifying. How do you practice applying biology? Well, that's what these sessions are about. So this session, let me make the slide do the slide thing. There's a lot of review in here. But one thing that the AP Bio team at Fiveable has talked about is, so these questions are only FRQ. So it's not like you can make a Quizlet, like what is the powerhouse of the cell? How does the cell make energy? How does it do this? Da, 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 da. Can't really do that. I mean, you need to know that stuff, but it's all going to be your open-ended answer. So what we have decided for the content of these free streams is going to revolve around labs. So these are traditional labs that you will complete at school. Sometimes you have, sometimes you haven't. It depends on the resources of your school and your teacher. Like, for example, I didn't give this lab to my students. We just ran out of time, and that was right before schools got closed for COVID-19. So I don't know. That's too much info. I see some questions going on in the chat. I will address those in just a second. Let me finish this tangent first. So, but in the future, all of the free streams concern labs because we're going to talk about those labs because FRQ questions, especially the ones that they will be giving from the descriptions I've read, are highly likely to be based on labs. So if you're familiar with the labs, it gives you a leg up so that you can answer a little bit faster because they love to use the old labs. And that way you already kind of know what you're talking about before you even start answering. Also, it just gives you a good idea of how to practice that application. So that's our goal. So let me address these questions real quick. Will the exams be proctored on College Board? No. What I expect is they will give you an app that you'll have to download on your iPhone or your computer or whatever, and you'll have to answer through that app. So I imagine that will like be to monitor your time and stuff. I'm not 100% sure, but that would be my guess, but they haven't released that information. No one's going to be proctoring it, but they have told us who are going to be readers, because I was supposed to be a reader this summer, that they will have measures in place to catch cheating. So like, don't be texting your friends and then turning in the same answers. How long is this going to take? The stream is about an hour max. And then isn't the test like 50 minutes, 45 minutes approximately? You will find the exam on the app or on AP Classroom. They will have something. I don't know what. To be perfectly honest, they may not know what yet. This is kind of an unprecedented situation. Like, the sky is falling. We don't really know what's going on, but they're working on it. And the important thing is they responded when AP students said, we still want to take our tests. So we're going to do the best we can. Okay. Okay. Any other questions? So many questions. If we go back to school, will we take the traditional test? No. If you go back to school, you may take it at school, but you'll still take it like online separately. And I mean, a lot of it just depends on your state. Okay. I'm going to go ahead and move on. If you have any more questions on the AP exam itself, let's hold those till the end, because I'll be honest, I have the attention span of a squirrel and I keep looking at that chat box. But let's hold the questions about like the logistics of the AP exam until the end, because I'm seeing a lot of questions be repeated also, and I don't want to waste anyone's time by repeating questions multiple times. So we're going to do that at the end. In the meantime, let's focus on content and making sure everyone gets what they need. So if you're already familiar with the exam logistics, then you can just leave at the end. Sound good? I'm going to just assume that sounds good. Here we go. So we're going to be talking here about heredity, gene expression, and gene expression regulation. That is because all of these things are going to play into the transformation lab, which, like I said, is a big lab in AP Bio. We're actually going to look at the procedures that College Board published on the transformation lab and talk about what kind of questions they may ask you on that lab. So, first things first, we're going to talk about heredity. This is the part where I'm going to drill you guys a little bit. I'm going to ask you guys some questions, so feel free to answer those questions as I ask them. For example, who was the guy who made up Punnett squares? $10 points to you guys. Great job. So Punnett squares were like the first practical application of heredity. They were made up or created by Gregor Mendel. Now, okay. Please do not use that sort of language. We do not need that. Okay, so okay, here we go. So Gregor Mendel was like the father of genetics before genetics was a thing. He did all this stuff like hundreds of years before they even discovered DNA. Who were the people who discovered DNA again? Who discovered DNA or the structure of DNA? The two people, yes, Sierra, there were two people. What were their names? Watson and Crick. Okay, very good. So Watson and Crick were the ones who came up with this or figured out the structure of DNA. Keep in mind, though, they did have assistance from Chargaff and Rosalind Franklin, but they're the ones who got all the recognition and blah, blah, blah. But in the meantime, when we talk about heredity, we talk very specifically about DNA and the traits it gives us. So we've got our double helix, and you take your genes. So a gene is just going to be a segment of that DNA. And then I like to think of them as like books, recipe books specifically. So if you've ever opened a recipe book, maybe you're like at your grandma's making cookies or something, you know, it's divided into chapters. It's got appetizers, entrees, soups, salads, desserts, okay? So it's kind of like each chapter is like a different chromosome. That's the way I think of it. And then each recipe in each chapter is like a gene. So you've got your chapters or your chromosomes, and then you've got your recipes, aka your genes. And so each gene has a recipe for how to make something about you. So for example, I have blue eyes. Hi, there they are. You can see them. And to make those blue eyes, that means I have a gene that contains a recipe that tells my body, this is how we make blue eyes. Okay? All of that is stored in my chromosomes. Okay? Now, here's the other deal. It's kind of tiny, but maybe you can see there's a little pedigree chart over here. Heredity is not just about like your individual genes. Heredity is going to be about your genes and how they can be passed on, or how you inherited them from your parents. If you saw my original stream on heredity, you would have seen pictures of my family because I look nothing like my family. I'm blonde. They are all not real blonde, but like I am blonde. It's just kind of like discolored right now. We're in quarantine. I can't get my hair done. But if you ever like see pictures of my parents, like they have dark hair, dark eyes, they're tan. I'm not. But then if you go further up my family tree and you see like my grandparents, yeah.
There's a severe thunderstorm coming this way with hail. It appears like it's going south of Jonesboro, but just want you and your students to know.
Okay, thank you. Sorry, guys. Apparently, we've got some severe weather about to come through my area. My husband just let me know. So if like my screen goes dark, that's why. But in the meantime, I'm going to keep teaching. Whoop, whoop. Oh, we'll be fine. Thank you so much for your concern, guys. Okay, so reinforce my walls. Y'all, I have like a tornado shelter in my bathroom. I'm going to be good. So let's focus on this stuff. Thank you so much for your concern, though. Okay, so where was I? We were talking about the genes and my eye color and my parents and all that stuff. How do my genes translate from this curly Q stuff in my cells to like the freckles on my face and the tone of my voice and my ability to hear? How does all of that stuff translate? Like, how do I get my genes to be physically expressed? Gene expression, RNA. Very good. Good, good, good. Y'all are so smart. Okay. So how do our genes make us? I hope y'all don't mind, but I show this to my students all the time. And like, I really needed everyone to have like a chill moment because I believe we all need that in these times. So we're going to watch The Little Bell and I. Hope that's okay with y'all.
Ever had someone say to you, it's in your genes? They weren't talking about your pants. Ha! No, they meant you were born that way. It's in your genes. Now, genes tell your cells what to do. Like, you're a stomach cell. You're a brain cell. You're going to have brown eyes and a big nose. Now, genes are made of DNA, deoxyribonucleic acid.
What?
Deoxyribonucleic acid.
What?
Deoxyribonucleic acid.
Okay.
And DNA molecules are very long molecules that have chemicals on them in a sequence, in a special order. Now, parts of DNA molecules are what we call genes. Now, DNA molecules are packed in your cells in something called chromosomes. Now, humans have 23 pairs of chromosomes. You get 23 from your mom and 23 from your dad. That's
46.
46.
Uh-huh.
Now, all plant and animal cells have chromosomes. They have different numbers depending on which plant or animal you're dealing with. DNA is pretty cool when you think about it. In fact, thinking's in your genes too.
Okay, so I know it wasn't like the most high quality video. But like, come on, everyone needs a little Bill Nye every now and then. Like, come on, seriously. So just wanted to do that. It's also just a really quick review to talk about how do our genes make us? Like, how do they make you, you? What is different about my DNA from my parents' DNA that we look so different? What is different about my DNA from my dog's DNA that makes her completely different? Like, how does all that work? You guys already answered that question because you're awesome by talking about gene expression. So we're going to review gene expression real quick. Gene expression is known by several different terms. So you can call it gene expression, protein synthesis, the central dogma. Any of those are completely acceptable. Central dogma is the coolest name. I agree. It just sounds like central dogma. Boom. That's the one I always use with my students, and they're always like, what's a dogma? Why is it central? And I'm just like, it sounds cool. Just roll with it. You can also call it gene expression or protein synthesis. Either one is fine. All right. So I've got a lot of different stuff rolled in here because like that's how I teach, by throwing a bunch of stuff on the board. And also I believe it helps people to have multiple things to look at and they can relate through whatever they want. So maybe, let me see if I can get my little pointer here. Maybe you like diagrams that show the whole process all at once. Maybe you like simplified diagrams. Maybe you like in-between diagrams, or maybe you like GIFs. I personally like animated motion pictures. So I use a lot of GIFs. So DNA, RNA, protein. We see that simplified right here. And then we can see in our next simplest model, you get this DNA. It gets transcribed, and what does it make in transcription? What does DNA become? mRNA, very good. What does the M stand for? Messenger, very good. And what is made in translation? Booyah, protein, very good. Yes, perfect. Okay, so that's good. And then if you look down here, you can see we've got a bunch of different enzymes that are involved in this process, okay? So when we start with that DNA, we need RNA polymerase. We need, I'll quit blocking my screen. And what is that RNA polymerase gonna do? Okay, please stop doing that. There it goes. What is RNA polymerase gonna do? Transcribe? Read and write mRNA, speed up translation, transcern? Huh? Catalyze the reaction. Okay, so grains of truth in each of those, but probably the biggest grain of truth is going to be where it reads and writes mRNA. So just like DNA polymerase, RNA polymerase is going to run down the DNA and be like, pew, 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 and it's going to make an RNA strand, and it's going to go pew, 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 and there's going to build that RNA, just like you can see in the GIF. No primers are needed. So I don't typically talk about the primers. There are primers involved, but I'll show y'all this. In the AP Bio Bible that they give you when you go to teacher school to teach AP Bio, it says you don't have to know the primers. So I don't address it just because people have enough stuff to memorize without adding on to that. But if your teacher taught you that too, that's great. What takes out the introns? Y'all tell me. What takes the introns out? Splicing. Okay, splicing is the process. What are the molecules that take it out? Spliceosomes. Very good. What book is this? You cannot buy this book on Amazon. They ship this to you whenever you become an approved AP Bio teacher. Like, you have to go through an audit and a training and stuff. But I mean, you can like find the contents of it on the internet. It's just like the curriculum plan. It tells you what you need to know and stuff. So this is what they give teachers. It's really boring. Like, don't pay money for it, please. Like, I promise you, your teacher has already taught you this stuff. If they taught you about primers, they taught you more than this. So, okay. Where was I? Oh, yes. Transcription and translation. So we can see the most detailed version of this on this one. We've got a cell. Transcription is happening here. But then before it can exit the nucleus, it has to go through post-transcription. What happens during that post-transcriptional processing? Turns to mRNA through splicing. And what happens when we splice it? Not oof. Exit. It cuts off the introns. Very good. And when it cuts out the introns, it leaves behind the exons. Very good. And so those exons get stuck together, and that becomes the mRNA that will actually be translated after that mRNA leaves the nucleus. Now, this example that I've got up here, is this a eukaryote or a prokaryote? Eukaryote. Very good. How do we know? How do we know it's a eukaryote? It's got a nucleus. Excellent. Is prokaryotic gene expression going to look the same? No, why not? It's a loop. Very good. The DNA is a loop instead of a long squiggly thing. Why else? No splicing. I don't know about don't have introns or don't have transcription. There is something that prokaryotes don't have, though. Nucleus. Very good. It's because they don't have a nucleus. So in eukaryotes, you have to do transcription, finish it all the way, then you can do translation. Prokaryotes can do transcription, translation all at once because they're not separated by a nucleus. Boop, boop. Okay, so let's take a look a little bit more at transcription. So transcription, you can see a really detailed GIF here. I found that on the internet. I was very proud of myself. But then we've got a diagram here, and you can see it's been opened. And once it's opened or unwound, we've got this RNA polymerase that's creating the RNA transcript. But before that RNA transcript can move on, you have to go and remove the introns. Now, to be clear, okay, listen carefully here. Depending on what protein you want to make, they may take out different introns, okay? When I was a kid, we had a lot of like sci-fi novels that talked about this. They would call it junk DNA. If you've ever heard of junk DNA, they like to talk about it in like the Maximum Ride books. Did anyone ever read those books? They were the books about the girl who like had wings and stuff. So there was this theory that humans all had junk DNA, which was like DNA you didn't use. It's kind of like that expression. They'll be like, you only use 10% of your brain, da, 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 da. If you've ever seen the movie Limitless, where they're like, you don't use all of your brain, but if you could, you'd be super smart. Not true. You use all of your brain. You just don't use it all at once. But back to that example, sci-fi used to be like filled with this idea of junk DNA. And like, if you could access the junk DNA, you'd get like superpowers. It was kind of like X-Men or whatever. And that was based on scientists knew there was certain DNA that we didn't use. And they were like, oh, it's junk DNA. It's superpowers. Ha ha ha. While don't get me wrong, I would love to be an X-Men and like have superpowers and stuff. Not how it works. What really happens, that junk DNA or those introns or whatever, you can remove different pieces at a time, kind of like different pieces of a puzzle or different Legos. And whenever you do that, you make a different mRNA transcript, and that transcript will make a different protein. That's why you have junk DNA. It gives you different codons. Very good, Japesh. Okay? Now, what is going to tell the DNA which protein it needs to make? That's going to be up to like transcription factors, which we're going to cover in just a minute. So translation, you're going to need a ribosome. The mRNA is going to leave the nucleus. It's going to float around, floaty, floaty, float. And it's going to have a poly A tail and a GTP cap. And that GTP cap, think of that as like the driver of the train. And he's going to be like, okay, here we go. And then it finds a ribosome. Meanwhile, the poly A tail is like the caboose. And he's just like, I'm here for the ride. You don't need to know all the details of that in this review. There's other videos of that. But in translation, that's where you're going to take that mRNA and you're going to take it and it's going to be read using tRNA to make the long string of protein. Ta-ta box, we're going to get there. Okay? And then you have a protein. So, yay. Okay. Any questions about gene expression so far? Please only type if you have a question. Like a legit question, because otherwise it fills up real fast. Although y'all's engagement is great. 5, 4, 3, 2, 1. Okay, I'm going to go ahead and move on, and we're going to take a look at gene expression regulation. Oh, but before we do that, we already kind of talked about this, but keep in mind, prokaryotic gene expression is going to be different from eukaryotic gene expression. It's not going to have a nucleus. The process occurs all at once, and there's going to be less DNA to transcribe. They also, I forgot to put this on there, but what is another difference between prokaryotes and eukaryotes, like between their organelles? Something else that's different. No, not mitochondria. They don't have membrane-bound organelles. They don't, but what do they have that you need for translation? They only have ribosomes, okay? Is there a difference between the ribosomes of prokaryotes and eukaryotes? They're free-floating. What else? The size. Very good. So prokaryotes, when they do transcription translation, no nucleus, so it happens all at once, less DNA, and they're a lot smaller. Okay, those are the big differences there I needed you to know. This is going to be relevant in just a minute when we talk about transformation. So now let's take a look at this gene expression regulation. So when we want to express our genes, when we want to use that junk DNA or whatever you want to call it, and try to diversify our genes. So something that I was told that kind of like blew my mind, like it exploded. I think we have like, can't remember if it's 10,000 or 100,000 genes, but you need like 250,000 proteins. So there used to be this thing called the one gene, one protein hypothesis. And the idea was that for every single gene, it makes a different protein. We have since debunked that because you can do alternative splicing, which is when you move different introns in and out. So now that's how we can make more proteins. But how does your DNA know, okay, we need this protein, no this protein, no this protein? Okay, so to do that, we have these transcription factors. Very good. The TATA box, it's going to need polymerase. It's going to need to be activated. So there's a gene that codes for an enhanced and it folds to it. Very good, Hayden. Promoters. Ooh, y'all are so smart. Okay, so we're going to do one more quick video. Here we go.
For RNA polymerase to successfully bind to a eukaryotic
Oh no, come back.
promoter and initiate transcription, a set of proteins called transcription factors must first assemble on the promoter. The assembly process begins upstream from the transcription start site, where proteins called basal factors bind to a short TATA sequence in the promoter. Other basal factor proteins then bind, eventually forming a full transcription factor complex able to capture the RNA polymerase. Basal factors are essential for transcription, but cannot by themselves increase or decrease its rate. A second set of transcription factors called coactivators link the basal factors with a third set of transcription factor proteins called activators. Activators are regulatory proteins that bind to sequences on DNA called enhancers. Enhancers are located at sites that are distant from the promoter. The interaction of activator proteins with transcription factor subunits increases the rate of transcription. Many enhancers, scattered around the chromosome, can bind different activators, which provide a variety of responses to various signals. When a second kind of regulatory protein called a repressor binds to a silencer sequence located adjacent to or overlapping an enhancer sequence, the corresponding activator is no longer able to bind to the DNA.
Okay, so very wordy there, and I will tell you, I don't think on the AP exam they're going to ask you, please describe the transcription initiation complex. They're not going to do that because that's all something you could get out of a textbook, and they don't give you points for anything that comes out of a textbook. So I do want you to understand it just because it will help you if you need to analyze and explain something later, but don't like put all your eggs in this complicated video basket. Like, don't. For those of you who are saying the PROG or FOROG, promoter repressor operon gene, that is super helpful. I've never used that with my students, and I am stealing that. Thank you so much. So when all this is going on, when you want to turn something on, if you will think back to like unit, I think it was unit 4 when we talked about like cell signaling and cell communication. So that was like autocrine, paracrine, juxtacrine, endocrine. There was also, what was the other thing it had? It had cell signaling, so like cascades of phosphorylations and things like that where they had to communicate and things like that. So this is a massive communication project to be like, okay, now do this, now do this, now do this. And you need the transcription factors and the TATA box, and it all has to fit together or it doesn't work. If you've ever seen like the gears on a clock, think of this as like a clock. If you miss a single gear, that clock's not going to work anymore. That being said, whenever we look at gene expression regulation in prokaryotes, it's going to be a little bit different because prokaryotes don't have the same mechanics to work with. That is, they don't have the same organelles as we talked about earlier. All they've got is ribosomes. That's it. So they have to use very specific molecules and enzymes in order to turn processes on or off. The big example of that we love to use, like the classic example that we love to use in biology, is the Lac Operon model. And so an operon is kind of like a light switch. So like I've got a ring light because I'm a millennial and I need good lighting. But when I hit my little switch, it goes off and then on, off and then on. An operon is just that. It's an off-on switch. But that off-on switch depends on what different things have been plugged into the switches. So the Lac Operon model makes 3 different genes, but the only way that you can make it is if you get rid of that repressor and make sure that you activate the actual gene. I will tell you, I would understand operons because they are very good for application. The lactose example, very good. So the Lac Operon is one of those that the light switch turns off when it doesn't need it. So for example, let's say that you guys are all home all day now. But does anyone here have a parent who's like, make sure you don't leave the lights on or the TV on and da 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 da? I have a husband who does that. But anyway, so this is the same concept. The cell is saying, hey, don't leave the operon on if we don't need it. So lactose is a sugar that the bacteria wants to break down. So it's only going to produce lactase when there's a bunch of lactose. Okay? So that's when it's going to start making the lactase, break down the lactose. But if there's no lactose, it's not going to make any lactase because that's a waste of time and energy. Okay? That's like leaving the lights on in a room that no one's in. Says all of our dads everywhere. Okay? Any questions about gene expression regulation before we move into the actual lab? What does the CAMP do? CAMP is kind of like a mini ATP. So CAMP, if I remember correctly, which I usually do, is going to be an ATP that only has 1 phosphate. And so it's a little energy charger, secondary messenger. Very good, Rudy. Okay. Any other questions? Why does it need to bend in the diagram? Why does what need to bend in the diagram? The DNA thingy? Oh, are you talking about here? Are you talking about in eukaryotic? Okay. So in this one, the reason it's bending is because all the different, oh my goodness, stop. All the different things that are here together. So they're kind of like magnets. Once those activators all line up together in those transcription factors, they will pull the DNA across because they've got to connect on an enhancer, which is up here, and on that ta-ta box. So it's got to bend around to connect them together. Does that make a little more sense?
Are those Jonesboro kids?
No.
Okay.
Yeah. Is it okay?
Yeah.
Okay.
No emergency, but you need to say something that they were Jonesboro.
Yeah, got it. All right. Sorry about that, guys. Another storm update. Looks like we're doing okay. So, whew. FBI open up. Yeah. Okay. So, one last surprise. Let's talk about viruses. The reason I talk about viruses is because this is the most applicable way to talk about transformation in terms of medicine beyond the basics of like the pGLO lab, which is what we're going to talk about. Obviously, viruses are a bit of a hot topic, so we're going to speed through these right now. So a virus looks like that little blob up there sometimes, or they can look like that little green and black thing. It just depends on what type of virus they are. Excuse me. But the point of a virus is they are very, very simple bacteriophages, very good vocabulary. Okay, so viruses have a very basic structure. In fact, most scientists don't even consider viruses living things because although they have their own genetic material, They don't usually have, what's it called? They don't have a cell membrane, at least not the kind that we have. They usually have a protein coat, or they can have a lipid coat, but they don't have like phospholipids like we have. Also, they don't have like any ribosomes. Like they don't have any organelles whatsoever. All they have is a little sac with their genetic material. So that's why they need a host, because they basically hop into the host cell and say, I'm the captain now. And they hijack it, and then they make the cell make viruses for them. Do viruses operate in the same way regardless of their structure? No. So some of them do what's called a lytic cycle. A lytic cycle is when they take over the cell, and this is kind of like, these are usually very fast viruses, so kind of like 24-hour stomach bug, or, you know, you get real sick real quick, but then you usually are fine. So what happens in those is they take over the cell, and they make viruses so fast that the cell will actually lyse or explode, and all the baby viruses pop out. Those don't usually last very long. They're different from lysogenic viruses, which actually hijack the cell, but then they go incognito. So picture like those memes where you see the people in the glasses with the fake eyebrows and the fake mustache and the fake nose. Like they go and they hide in the cell basically. But the way they hide, very good retroviruses, they actually take DNA, so a viral genome, and they will pop it in and make it DNA that like integrates in your DNA. That can be the incubation period for some things. I am not a doctor, so I don't want to comment on if that's the incubation period, because I'm not 100% sure. It's different because like what I'm talking about with lysogenic viruses, I'm talking about AIDS and HIV. And this is when a person can have the HIV virus in their system, but not necessarily know it, because the virus will basically just kind of sit there for a while, depending person to person, before it starts taking over the host cell and replicating and doing stuff like that. Incubation period, I would assume the incubation period is more the virus is already replicating. It just hasn't started presenting symptoms yet. Yes, so it's dormant when it's just sitting there. Very good, Rachel. Okay. That's a quick rundown of the biology of viruses. Does anyone else have any other questions? The one thing that I will throw in, so these viral genomes, if they can put themselves in our DNA, so that's like in humans, which they can, HIV is the example of that. There are scientists who are working on taking advantage of that and removing the HIV part, just like snapping it out, but then using that in order to insert new genes into people's DNA. So for example, they're currently working on trials for sickle cell anemia. So like that would replace the bad gene someone has for sickle cell, where they would then have the gene to make the proper blood cells. And it kind of ties into this transformation lab. The transformation lab we're looking at those with bacteria. So it's a gene therapy, very good. So it's a little bit more intense, obviously, with gene therapy. But bacterial transformation is like the kindergarten version of it. And that's why it's so applicable to AP Bio students. So without further ado, let's go talk about transformation. Da-da-da. Okay. One more thing, because I keep forgetting what's on my slides because I'm blonde. Sorry. So you've got an HIV virus here. And what HIV does is it does reverse transcriptase. HIV has RNA in it, okay? But that RNA goes through reverse transcriptase. So instead of becoming RNA into protein, it goes RNA into what? In reverse transcriptase, what does the RNA become? DNA. Very good, Lee. Very good, Jerry. All you got others. There's so many of you. Is heterozygous for sickle cell make you better against malaria? Yes. Moving on. So once HIV turns it into DNA, it then injects, you can see it here, into the person's cells. It attaches and then gets taken into the cell because the cell's like, oh boy, a snack. And that's because our cells don't have receptors to respond to it. But then the HIV DNA will actually go into the nucleus and then embed itself into the human DNA. Once it does that, then every time that DNA goes through central dogma, it will pass it on and start making new HIV viruses. That's why it's such a problem and why you can't cure it. It's also probably why it takes a while for some people to show symptoms, because it's not like the DNA embeds in the same place every time. It's very random where it snaps in. And so it just has to wait until it's eventually transcribed. So let's talk a little bit about the transformation lab, because we only have 15 minutes left and I'm supposed to be okay from a thunderstorm, but like, just want to make sure that my computer doesn't go dark because I keep hearing lightning. I'm okay though, I promise. So in the transformation lab, why do I have Homer Simpson with radioactive glow-in-the-dark stuff? Magic, yes. Talking about the transformation lab, why do I have glow-in-the-dark stuff? Radioactive isotopes, the glow gene, very good. Very good, Lee, you win, bioluminescence. Okay, so in the transformation lab, if you do the one that like your teachers give you, which real quick poll, I'm gonna ask a question. How many people have done the transformation lab at school? Okay, so I just asked a question. If you guys will do me a favor and go answer, so you can say yes, no, yes, no, yes, no, that sort of thing. That would be super helpful. La, 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 la. Okay. Just gives me an idea of like how many people have done it. Well, then it went away. Did I break it? Oh, there it goes. I broke it. Ha, sorry. Okay, one person has said no. Okay, there you go. Go ahead and start answering, y'all. Sorry. I apparently am really bad with technology today. Okay, it looks like most of you have not, in fact, done it. That is totally okay. It's a really, really, really like time sensitive and time using lab. It takes a lot of time because you have to like wait and let the bacteria grow. Then you have to do the lab. Then you have to wait and let the bacteria grow more. So if you haven't done it, it's totally okay. That's why we're here talking about it. So in the transformation lab, the goal is to make glow-in-the-dark bacteria. It does a transformation, kind of like that bippity-boppity boop thing that we keep seeing on TikTok. That's why I have the Cinderella gif there. But the goal is to take bacteria and you make it glow in the dark. The reason we make it glow in the dark is because, this is super cool, that makes it resistant to antibiotics. Obviously, antibiotic resistance is like not a good thing, but in controlled situations where you're going to kill the bacteria later with like bleach, it's okay. So here's what happens. It's a lot like a virus, which is why I tell my students about it. So in transformation, what you've got is a bacteria. We only use bacteria. We typically use E. coli, and those E. coli are, there are multiple strains of E. coli. This isn't like the kind that like makes you sick, like on spinach or something. This is a harmless kind, apparently, because we order it and pay a lot of money to a company for it. But so what happens is that E. coli, we expose it to DNA fragments or to a plasmid, and that plasmid or that DNA will actually get uptaken into the DNA. And then it will either get taken into the bacteriophagous DNA, or it may not. Or it may just sit there as a plasmid. Either or. Now, that's the natural reaction that is just like out there happening in nature. Do I have TikTok? No. So this is the natural reaction that's just happening in nature. But if we want to do cool stuff like make things glow in the dark, oh, it's so cool. Okay. So I know that picture's a little blurry. I'm sorry, it didn't like blow up well. Main thing I need you to know, especially for those of you who haven't done this before. Yes, you need to heat shock it. Very good, Jenny. Okay, so you start with a little petri dish that your teacher is gonna put a very specific agar on there. And it's gonna be a type of agar that E. coli like because they eat the agar and that's what keeps them alive. Then you're gonna have these two little tubes. They're called microcentrifuge tubes, okay? And inside those, you're going to have different substances, okay? There's going to be one that has a plasmid, another that doesn't have a plasmid. That plasmid is the pGLO. You put it in a blender and it's like, you're thinking of a different one with the blender, okay? So the pGLO plasmid, okay, what you've got is it's a loop of DNA, because remember, bacteria can have DNA loops, okay? And it's got 3 separate regions. It starts at the origin, that's the beginning part of replication. It's got BLA, which is beta-lactamase, and that's going to make it resistant to ampicillin. Ampicillin is an antibiotic that would kill that E. coli. It's got GFP, that's, green fluorescent protein. That's the part that makes it glow in the dark. Spooky, spooky. And then it's got ARA-C, or arabinose. It's an arabinose regulator protein, and that regulates the GFP expression. That is, it turns GFP off and on, off and on. Fun fact, where does that GFP come from, that green fluorescent protein? Where does GFP come from? I'll give you a hint. In Finding Nemo, they are called squishies. Jellyfish, very good. So that GFP comes from jellyfish, but here's the deal. Jellyfish are eukaryotic. Bacteria are prokaryotic. So that ARA-C is going to be involved in cows, yeah. So that ARA-C is going to be responsible for turning on the GFP, even though it's in a prokaryote instead of a eukaryote. So at the end of all of this, we're going to go through the procedure in a minute. These are the results that you're supposed to get, to be clear. So you're going to have 2 of these tubes, these microcentrifuge tubes, okay? And one of them is going to say plus, and one is going to say minus. The plus means it has the pGLO plasmid. The minus means it does not have the pGLO plasmid. That's going to be your control. So when we see here it says minus DNA, that means minus the pGLO. When it says plus DNA, that means it has the pGLO. So here, when we have the pGLO, go away, you annoying thing. Okay. So when we put pGLO and AMP, or ampicillin, unfortunately, that ampicillin kills the bacteria, and that prevents it from glowing and doing its thing. Can't uptake the plasmid. But if we put ARA, ARA, down here at the bottom, that ARA, remember, is going to be that arabinose regulator that's going to turn on pGLO. It's also going to turn on that ampicillin resistance. And that's what's going to make this thing start growing like crazy. Now, if we have DNA with, or if we have the bacteria with no plasmid, it can still grow a colony. But if we have no plasmid and ampicillin, it's not going to grow anything because the ampicillin is going to kill all of the bacteria. A lot going on there, I know. So we've got like 6 minutes left. Let's take a look at the actual procedure that they would ask you to do here. But before I do that, I do want to remind y'all why I'm doing this, okay? How is this relevant on the AP test? There's 3 reasons. You do need to understand the procedure of how transformation works, because if you don't, you won't be able to apply it to other projects. And what they're going to ask you, if they ask you about it on the AP test, is they're going to be like, okay, here's transformation. It's so cool. How could we use this to better humanity and stuff? Glow-in-the-dark pigs. Very good. You need to understand so that you can apply it to other stuff. You need to be able to interpret the lab results. So they may give you a picture of like these plates of bacteria, and they may ask you, hey, what's going on there? And you need to be able to tell them, okay? You just need to know how the process works. You don't need to have every little detail memorized because this is more of a broad understanding so you can apply it, but you need to know how it's working. And so, real quick, I'd like to give you all a little tip. You can Google, I'm going to stop share real quick. Dun, dun. Okay, so I'm going to open a new tab. Dun, dun, dun. I think I'm about to have to go though. I'm really sorry. So I've got a new tab here, and we're going to Google AP Biology Transformation Lab PDF. These are publicly available on College Board. Okay, these are the lab guides that teachers use. I'm not here to tell you how to get answers and stuff. What I'm here to show you is here's the lab guide. Okay, the reason that I am showing you the lab guide, yes, the slideshow will be shared with you. The reason that I am showing you the lab guide is because, like I said, I strongly believe that the free response questions will be concerned with the lab. So if you already understand the lab procedures, so you can go down here to like general procedures and stuff like that and materials, then you will have a much easier time answering the questions, okay? And that will make it a lot easier for you to be able to answer quickly, because you only have max 25 minutes on each question, 15 minutes on the other question. That way then you can move on and do your thing. Okay. Does anyone have any questions? You got me for like 3 minutes maybe. I am good. So far it's just like lots of thunder and lightning. I just don't want to lose power on the middle end, y'all. I strongly believe that at least one of the FRQs will be focused on labs. Olivia, I don't know if both of them will be, but I strongly believe at least one of them will, and the other will be some scientific phenomenon. If they tell you to apply and use the insulin example, will they count it? They will count the application, not the content. How to analyze karyotypes? No, I think that's in the curriculum guide. But I mean, even if it is, it's just going to be like, what is wrong with this karyotype? And then you would say, oh, this one has 3 chromosomes instead of 1, or this has 1 chromosome instead of whatever. Brief overview of what the transformation lab can show. So that's what those plates were, Rachel. So the 4 plates where one was glowing in the dark and the other 3, those are the types of results you would be expected to analyze. But is what a source from the book? It's 45 minutes in total, right? Yes, 25 minutes for one question, 15 minutes for another question with like 10 minutes to upload stuff. Will they be focused on labs that are within the curriculum? That's what they've done in the past. Okay. So what's the benefit of that lab supposed to show? That lab is supposed to show you how you can use pGLO, or you can take a plasmid and you can integrate it into another thing of DNA to change the genes that it expresses. So you've got genes, like we said in the very beginning, like we said with Bill Nye, you've got genes. I have genes that make my eyes blue and give me freckles and all sorts of other stuff. But how do those genes go from little squiggly DNA to being expressed? So the point of this lab is that I can add new DNA, and that makes a new trait to be expressed. That makes things glow in the dark or resistant to antibiotics. Does that make sense, Olivia? Okay. All right, y'all. I'm getting like weather updates, so I'm gonna log off. I apologize for logging off a minute early, but I think I need to like unplug my computer. I'm really sorry. So, but I think I'm gonna be okay. So it's gonna be all right. But guys, y'all were awesome. I loved how much y'all were collaborating and doing all this stuff. Let me share these slides with you before I go. And then can view. All right. Bye, y'all. And FRQ suggested